everyone, welcome back to my Coursera class, Biochemical Principles of Energy Metabolism. In the previous previous session 3 for week 2 is about carbohydrate digestion. So far we have reached the step of carbohydrate, let's say glucose degradation throughout those glucose polymer degraded like throughout amylase actions, and now finally glucose monomers produced. And you faced many different types of enzymes. So obviously next step is how glucose can be absorbed into our body. So instead of explaining the glucose or carbohydrate absorption process, I'm going to have a review session for enzyme. Okay, so what is enzyme? I think you are going to see many different types of enzymes throughout this class. So it's going to be very important for you to have clear and solid foundation for enzyme function. So what is enzyme? Enzyme is a substance that can act as a catalyst. In particular, catalyst in living organisms. That is called enzyme. Okay, so what's the main point or main action of enzyme in biochemical reaction? Just look at this graph, and let's say that this is a simple biochemical reaction. Carbon dioxide and water, product is bicarbonate. So free energy reactants like this high and products like this low, and throughout those reactions, depending on the presence of enzyme, the rate of this reaction can be hugely changed. The point is, without enzyme, this reactant should go through this high energy status and then relieved into product. So without enzyme, this amount of energy change throughout this chemical transformation is called activation energy. Activation energy is that high. The thing is, under the condition of the support of enzyme, this activation energy can be lowered. So that means by reducing the activation energy, that means the enzymes can dramatically increase the rate of biochemical reaction. So there are over 5,000 different types of biochemical reactions available in the living system, and enzymes are the major player in terms of trigger and facilitate the biochemical reactions. So as you can see, this diagram, enzyme binds to substrate, and enzyme substrate complex. So this enzyme assist and facilitate the biochemical transformation of substrate into product. And this catalytic step, I'm not going to go into very detail. The kcat, catalytic power, kcat, can be hugely increased. And most enzymes are proteins, but few enzymes are known to be RNA-based. And this is quite important because when the biochemists and biologists for the first time identified RNA-based enzyme, and we are really surprised. And then this is the Nobel Prize topic of a decade ago. Okay, and enzyme, their catalytic activity, how can be regulated? There are many important factors. I'm going to list up one by one. First one is temperature. And you know that, right? The temperature overall can increase the rate of reaction. Why is that? When temperature rises, the substrate molecules can have more kinetic energy, right? And that increases the chances of a successful molecular interactions. And finally, the rate of biochemical reactions. The thing is, enzymes' catalytic activity requires optimal temperature. And our body temperature is 37.5 degrees in Celsius, and that's the optimal temperature for the enzymes in our cells. And this is diagram. Around 37 degrees is the highest activity of enzymes. The second factor is pH, in other words, acidity. So each enzyme, it's a protein-based enzyme or even RNA, whatever, they can operate in a small range of pH. And there is optimal pH condition for the best performance of enzymes. Okay, why is that? Because the pH acidity, that means proton concentration, can affect the molecular interactions around enzyme and substrate. Therefore, enzyme functions can be changed. So some enzymes, this is pH 7, pH 0, and pH 14. Some enzymes are really active around pH 7. Some enzymes are really active under acidic condition or in basic condition. Some examples over here. Like amylase, we studied in the previous session. Amylase can degrade carbohydrate into smaller pieces, but the optimal pH is around 7. And what about maltase? Intestinal maltase, also they can do their own job around near neutral pH. But in the case of pepsin, pepsin is the enzyme for protein degradation into small amino acid or smaller chain. Okay, and this pepsin degradation occurs inside your stomach. And in the stomach, there is hydrochloric acid, and that very acidic condition, pepsin can be fully activated and can function in degrading a protein nutrient into amino acid or smaller polypeptide. Number 3 is cofactor. Some enzymes require coenzymes, that means small organic compound or mineral cofactors, zinc, sodium or potassium, those kind of mineral-based cofactors. When those cofactors or coenzymes, they directly bind to the active site of the target enzyme, then that enzyme can be fully activated. And these small organic molecules for coenzymes are indeed, many cases, derived from vitamins. Ultimately, that means from the diet. And other coenzymes or cofactors like metals, many metals, also supposed to be supplied from the environment, I mean from the food. So again, the concept of cofactor is some enzymes absolutely require cofactor, maybe some organic molecules or some minerals, and then form a hollow enzyme, and this is functional. And this protein structure diagram, this is carbonic anhydrase. You don't need to remember the structure of this enzyme. The thing is, in the middle, you are looking at the silver ball. This is zinc. Without zinc, this enzyme cannot be active. That cofactor is an absolute factor for full activity of target enzyme. Number 4 is modification. So protein is modified. The nature of modification is covalent modification. When protein-based enzymes are modified, chemically modified, their activity can be changed. That modification can occur on top of specific amino acid side chain in the middle, or maybe extreme C terminus or extreme N terminus. But the point is, when the protein enzyme is chemically modified throughout covalent bond, that is called post-translational modification. Throughout this modification, this enzyme activity can be increased or decreased. This is another layer of enzyme activity regulation. So best well-known example is phosphorylation. When enzyme is phosphorylated, then enzyme function can be regulated. So this is Nobel Prize topic diagram. So there is an enzyme, glycogen phosphorylase. When this enzyme is phosphate attached, throughout those phosphate covalent attachment, covalent modification, then this enzyme activity can be fully manifested in an active state. So this enzyme activity can be regulated by simple phosphate attachment or detachment throughout phosphatase or kinase. And this event, biochemical event, is one of the key features in terms of the glycogen reutilization for energy sources. And there are different types of post-translational modification are identified. And among those 4 different factors, temperature, pH, cofactors, and post-translational modification can regulate enzyme activity. So in that sense, I'm going to introduce one medical symptom called lactose intolerance. We are looking at the world map showing that many human beings are affected by this symptom called lactose intolerance. So basically, lactose intolerance is caused by the reduced capacity to digest lactose. In particular, lactose is highly enriched in the dairy product like milk. So this lactase is very, very important intestinal digestive enzyme. When this lactase is not optimally expressed and function, lactose cannot be degraded into glucose and galactose. In that case, abdominal pain and bloating and in severe cases diarrhea and nausea can be developed. So that's why some people consume lactose-free or low lactose dairy product. And in some severe cases, the medical doctors prescribe lactase tablets. That enzyme can be directly used to fully digest the lactose ingredient from the dairy product. So enzymes sometimes nicely inhibited and those inhibitors are basically the biochemical basis of drug development. There are 2 types of enzyme inhibitors available. Number 1 is competitive inhibitor. The point is, enzyme can bind to substrate, and this area is called active site. When some inhibitory molecules directly compete with the substrate in terms of taking this active site position, that is competitive inhibitor. But on the other case, non-competitive inhibitor does not compete this active site with substrate. Rather, there is another distinct area of inhibitor binding site. The thing is, when this non-competitive inhibitor can bind to target enzyme, and this enzyme, the structure and function or the substrate binding can be severely and negatively affected. Thus, the non-competitive inhibitor treatment can reduce the maximal catalytic activity of target enzyme. So today's session is about enzyme. Conceptually, it's very clear. It's a biocatalyst and absolutely essential for metabolic reactions. in terms of reducing activation energy by elevating catalytic steps by forming a complex with substrate.